Hello, I'm your host, Ray Dogum, and welcome to VibeCast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel important drug development. Vibe Bio seeks to find every cure for every community, and we think big as no one should be left behind in the pursuit of living a healthy, happy, and productive life, free from disease. Collectively, we have the skills, we have the technology, and we have the passion. We now need the community catalyst to put that all together. That's Vibe. We see a future where communities of biopharma experts and patients collaborate to identify high potential medicines and have the ability to access capital on demand to actually develop them. The VibeCast is our weekly informational podcast where we explore some of the hottest topics in drug development and technology innovation with some of the dynamic people that make up the Vibe community. Join us to learn, imagine, question, and help us identify and develop solutions together. Our guest today is Dr. Mo Jain. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, biomedicine, engineering, computational biology, and mass spec-based metabolics. Dr. Jain founded Sapient in 2021 as a spinout from his laboratory in UC San Diego to expand upon the mission of accelerating human discovery and drug development. Dr. Jain, how are you today? Thank you for joining me.
Thank you so much, Ray. It's a pleasure to be here, and I appreciate that vibe intro. That very much resonates.
Thank you. And is Dr. Jain okay, or should I call you Mo?
Mo is fine.
Awesome.
As I mentioned, Dr. Jain gives me PTSD. My pager's going to go off. I'm going to have to run somewhere. So Mo is great. Awesome.
Would you like to briefly also introduce yourself and sort of tell us what led you down this career path?
Yeah, absolutely. Happy to, Ray. So I've had something of a circuitous path through life where I'm still trying to figure out what I want to do when I grow up. And so I started as an MD, PhD, was very much enamored by clinical medicine, and to this day, absolutely love taking care of patients and helping. And as I was going through my training in the Boston programs, and then really learning how to be a doctor, there was a very frequent question that came up from patients. You know, Boston's a very educated place. And so you would have these professors who are admitted to the hospital and they'd ask, well, why did I develop this disease? And you would start telling them about sort of the pathophysiology, what's in the medical textbooks, trying to sort of make it simplified. And they would oftentimes crinkle their forehead. And they would say, none of that makes any sense. You really don't understand why someone develops disease. And that's the honest truth. We really still, to this day, don't understand much about why one individual may develop a disease and then one individual stays healthy. Now, we know certain risk factors, smoking is bad for you and eating well is good for you. But beyond that, for most individuals, that fall in the middle range where they've lived their life in a reasonably healthy way, it's hard to determine why someone develops a disease state versus not. And so I became very interested in that idea of really trying to decipher root cause of disease. And through that training, really spent a lot of time in mass spectrometry, large data systems analysis, became a professor in Boston, then ultimately moved my laboratory here to the University of California, where we developed some very specialized technologies that allow us to be able to measure things in human blood, to peer in a way that we had never done before. And as part of that process, then we ultimately decided to spin out Sapient to make available to the larger scientific community these technologies. And here I am today.
For the audience who might not be familiar with mass spec, and I know that's something that's really important to just briefly explain, can you take a few seconds to just uncover or unpack what that means and how it applies to medicine?
Absolutely. So I'll take a step back and even start with why even mass spec? Why do we care about this? And as I mentioned earlier, we really don't understand why someone may be healthy or not healthy over the course of their life. And this really breaks down into two components. There's a genetic underpinning that you're born with that starts in the moment of conception. It's what you inherit from your mother and father. And then there's the way you live your life over the next 100 years. Everything you eat, drink, smell, smoke, everything you're exposed to, everything the way your organ systems work internally. Now, that genetic underpinning, we've gotten very good at analyzing. This is what sequencing has done for us. This is what genomics and that whole genomic revolution of the last 20 years has done. The challenge is this, that even if we sequence every person on the planet, even if we line every single person up, we are able to understand their genome fully, we can begin to estimate, well, how much of human disease will we be able to explain? And we call this population attributable risk. And it turns out the number is somewhere in the order of about 20% or less. Which is absolutely fascinating. So the genome, while important, only provides a very small portion of the story. And we know this. We know the way you live your life has a huge impact on how healthy you're going to be over the course of your life. And none of that is captured in genomic information. So we became very interested in that other 80%. Everything that, again, comes from what you eat, drink, smell, smoke, the microbes in your gut, how you live your existence, those dynamic measures that very much influence health and disease. And that information is not encoded, again, in genetic sequence, but is encoded largely in small molecule chemistry. So to give you an example, Ray, you had breakfast this morning, you ate something, it got broken down in your gut into small molecules that travels through your blood. And because you are exceedingly healthy, those are all good things that have a positive impact on your cells and your organs. Now, the whole idea is if we can assay those small molecules in blood, we can begin to understand how your body is reacting to certain diets, how your liver works, how your gut works, how your fat cell works, how your brain works, et cetera, et cetera. And that becomes the real information source. Now, to answer your question, what mass spectrometry is it's a very large device that allows us to take very complex specimens like a human blood sample that's made up of tens of thousands of things and measure the abundance of those factors that are in blood.
Exactly. And can you provide some examples of what you measure? And basically it's anything, any kind of chemistry really, but just so that the audience understands like how it applies to medicine. Can you give some examples?
Yeah, absolutely. So what we're specifically measuring are what are called metabolites and lipids. And these are a class of molecules that are organic. So they're made up of carbon, hydrogen, and oxygen. They fall within a particular size range and a particular chemistry range. And those are the molecules that we're typically assaying through these types of devices. Now, when you go to the doctor's office every year, they draw 2 tubes of blood as part of your annual physical. And in that tube of blood, and those 2 tubes of blood, we're measuring about 12 different things, 6 of which are metabolites or lipids. That includes cholesterol, triglycerides, glucose, creatinine, urea, nitrogen, et cetera. So these are sort of some examples of the ones that we measure now as part of clinical testing. The challenge is that there's over 12,000 things that fall within this chemical class that are in your blood. So we're assaying less than 0.1% of them, which means there's many, many, many more molecules in blood that can be used to help us understand health and disease. Now, what we've done at Sapient is we've developed technologies that allow us to assay those tens of thousands of markers that are in human blood, and to do this hundreds of times faster than was previously possible.
So let's jump towards or jump back a few years when you started Sapient. What was your mindset at that time? And this was probably peak COVID time. So what drove you to start the company?
It was sheer boredom. Really. So, this was an experiment, and I'll honestly admit, most of the best things in life are oftentimes not necessarily deliberate. They're happenstance that you see as opportunity. And that was certainly the case here. And so we were developing these technologies for the last decade at the University of California as part of my academic laboratory, essentially taking mass spectrometers apart and making them go faster and faster, building software applications on the back end to analyze these data. And a number of organizations were coming to us and saying, hey, we'd like to access these technologies, large pharma companies, big foundations, international organizations. And they were saying, help us. We have these 10,000 samples. We have these 50,000 samples. What can you do to help us measure these? And as we were doing this, we said, there's some of these relationships that just don't make sense to bring through the university for intellectual property reasons or just complicated contracting. And the academic public universities are not always the easiest place to interact with industry. So as you said, in the height of COVID, we got this brilliant idea to spin out this organization. And again, it was largely done out of the fact that there was a lockdown. The university was essentially closed. And I was tired of sitting at home playing with the dog and kids and spouse, and there was really nothing to do. And so we said, let's try to learn something and we'll spin this out. And it was meant to be a very small organization when we spun it out. I was never sort of intending to leave my academic post, which I absolutely loved. I loved my laboratory. I loved my students. I loved my teaching and my clinical work. And we decided to do this and keep it small. And lo and behold, it somewhat exploded out of the gate and continued to grow quite rapidly. And as part of that, I had to leave and come on to lead the organization.
Are you still practicing medicine?
Not anymore. Unfortunately, I've had to put it on pause. There's just so many hours in the day between work and family. And so that was the one that had to go by the wayside. I'm still involved with a number of trainees who are going through clinical practice, fellows and whatnot. And I try to take time to provide advice and work with them when I can. But unfortunately, I just don't have the opportunity to practice clinical medicine anymore.
Totally understand that. So now thinking about the technology that you're developing and have developed, what is the actual significance of this biomarker discovery technology? And how does it apply to accelerating therapeutic research?
Yeah, exactly. So again, the way medicine works, the crux of medicine is being able to diagnose a disease and then treat that disease with a medication that's appropriate for that specific disease state. And that's the fundamental principle of how we go about sort of working in medicine. Now, diagnostics, as I mentioned, have evolved tremendously, and you could argue that the greatest benefit in human survival have come over the last 50 years because we've gotten much better at diagnosing disease in an earlier stage. But there's still quite a ways to go before we're perfect in this. And again, one of the real sort of ways in which we can begin to do this is by measuring these thousands of markers in blood, which we're measuring a dozen of which now. There's over 1,000-fold more markers in blood that are not currently being assayed. And the whole idea is if we're measuring 12,000 versus 12, we can tell you a lot more about your health and disease. We can understand when you're going to develop a disease years in advance. Disease, we know, is not a singular process, but is a decades-long process for most adult onset diseases, whether it be heart disease, cancer, neurodegeneration, liver disease, et cetera. These things occur over years, not over months or days. And so being able to diagnose these at these earliest time points is critical. Once we diagnose disease, being able to understand what's the ideal medication is the second question. Now, the way we treat disease now, for the most part, is as a singular entity. We treat all diabetics the same. We treat heart disease patients the same, for the most part. Now, the challenge we know is that there is great degree of heterogeneity in each of these populations. What we call diabetes probably represents 5 to 10 different diseases, all of which result in metabolic imbalance, all of which have different paths from which a person goes from a normal state to a disease state. Now, treating all of these as a singular homogeneous grouping is not ideal, as you can imagine. And this is why over half the people that receive a particular medication don't actually respond to it, simply because we're not aligning the individual, their disease, and a particular therapeutic. Again, this is where these markers can really, really transform medicine. We've seen this in the oncology space with genomics, and now it's time to sort of apply the same concept of being able to intensively interrogate disease, understand how you went from the normal to the abnormal state, and best align that with a particular therapeutic.
So let's talk about innovation in metabolics, metabolomics, I guess the study of metabolites and metabolites and different types of solutes in your blood and body, et cetera. So how has that changed in the last 20 years?
Yeah, so the idea of being able to understand the individual through the measurement of these molecules is not new. This concept has been around for decades. Mm-hmm. The challenge is, how do we actually go about practically doing it? And if there's thousands of things in blood, we have to be able to measure this in tens of thousands of people in order to really understand this is the key marker that's going to tell me Ray is going to be healthy for the next 100 years, or this is the marker that tells me that I may be at risk for developing disease over the next 10 years. And so that's always been the challenge. The technology has advanced tremendously over the last decade or so with what's called high resolution instruments. We can now measure things much, much more precisely. The challenge has always been that it's still quite slow. It takes hundreds of minutes to analyze a particular blood sample. And so what we worked to do over the last decade, and now at Sapient, was to take these instruments and speed them up greatly. And the rough back of the envelope calculation is we had to go about 100 to 500 times faster for this to really be deployed across large populations, to be able to analyze tens of thousands of samples at a given time. And so that's precisely what we started doing. And we developed hardware that allows us to go much, much quicker. We developed software that allows us to handle the data, the very complex information that emerges very quickly. And to give you an idea what that means now, Ray, we could take a biological specimen, whether it's blood, urine, CSF, your tears, a skin biopsy, whatever the case it may be. And we can measure somewhere in the order of about 15,000 of these small metabolites and lipids in that biological specimen. And we do that in about 53 seconds per sample. And that's what allows us to go much, much faster, to deploy this across large populations now and be able to say, from these 15,000 things or 12,000 things that are in Ray's blood, this is the one key marker.
Yeah, that's a huge step forward in our technology and what we can do. And, you know, this idea of scaling diagnostics so effectively, do you find that there will be any physical constraints, like in the laws of physics, that withhold you from reaching even faster diagnostics?
Yeah, it's a great question. So unfortunately, it's not the laws of physics here that are constraining. Certainly, there's some theoretical limitations, and I suspect we can get a little bit faster, but we're hitting up towards sort of the peak of what mass spectrometry as a technology as it currently exists can do. Where the constraints really lie is in human bias and human sort of culture, and that medicine, given the fact that you're taking care of people, involves health, it's people's lives. It's a very slow evolving process. There's regulatory constraints. The way we think about disease and diagnosis, the way we're trained even is very much based upon empiric information from the past. And we try to do things in a very methodical, slow way. Now, that means the evolution and the innovation in the medical space has typically been quite a bit slower than what we see in the tech space or in astrophysics or in these other areas where these same regulatory hurdles don't exist. Now, I think regulation is a good thing. Don't get me wrong. It's not a matter of saying we should limit regulation, but that turns out to be the biggest constraint. Taking new ideas and having adoption by the wider medical community after you've proven, in fact, the value of that idea is really the biggest limitation here.
If you were to sort of wave a magic wand and adjust some regulations that you currently face, what would be some of the initiatives or what would be some of the changes you'd make?
Yeah, it's a good question, Ray. I'm far from a regulatory expert, so I'm probably not the best one to answer that per se. What I can say, though, is where I think we'll be a decade from now is that it'll be a very, very different scenario where you'll be going to your doctor's office. We'll be routinely performing very complex genetic analysis as well as biomarker analysis using these types of markers that we've talked about, these metabolites, lipids, proteins, other measurements in blood. Again, not measuring a dozen things, but measuring hundreds of them. And from that, be able to provide a much more holistic view of your health state, your disease risk for dozens of diseases years in advance, as well as be able to provide far greater insight into what may be the ideal medication for your specific risk. And I suspect that this is where we're going to see huge changes in just the practice of medicine over the next decade or so. Now, components of this will involve AI and other sort of more advanced sort of statistical approaches. And again, I'm far from an expert in that space. And I'm still not clear to me exactly how that's going to impact the practice of medicine, but I do firmly believe that we'll be measuring more things, and where we're going to see massive advances over the next decade is going to be in advanced diagnostics.
Yeah, and I think taking those discoveries and understandings of the nature of humans and then translating that into therapies and treatments are going to be quite interesting, especially with cell therapies coming out and different types of other, modalities for treatments. That's really exciting stuff. And I think that it's changing so fast, so it's hard to predict. We're talking about 10 years. Seems like a short time, but actually so much can change. Now I want to ask you, switch gears a little bit, talk more about your funding and sort of like how you were able to collaborate with the Bill and Melinda Gates Foundation. And can you just walk us through like how you got introduced to them and what your work entails with them?
Yeah, absolutely. So as I mentioned, when we were academics and as we were developing these technologies, a number of organizations were coming to us and saying, hey, we're doing these clinical trials, we're doing these studies, help us understand disease, help us develop new diagnostics, help understand why individuals respond to our drug versus didn't respond to our drug. And one of those organizations was the Bill and Melinda Gates Foundation. Now, this is an incredible organization whose goal it is to transform life as we know it, not only medical care, but life in the developing world, what we call low and medium income countries. That represents the majority of the world's population, as you know. And there, in certain regions, the issues and the constraints are far different. Medicine is quite decentralized, meaning that there's not access to medical care for the majority of the world's population within 10 miles of their home. And so the way we think about medicine there is quite different. And one of the areas that the Gates Foundation has really invested quite a bit of effort in is understanding health from the earliest time points. So no longer are they focused on, I don't want to speak for them, but my understanding of how they think about it is that no longer is diarrheal illness, for instance, the major cause of early childhood mortality in the developing world, but rather it's issues that occur at the earliest stages of life during pregnancy that predispose children for having trouble in early mortality. And so they've made massive investments in trying to understand how they can optimize health and maternal health and childhood health in the earliest days of life. And that's really an incredible sort of mission. I could say personally, when my wife went through pregnancy, and we have twins, and so we went through this, and we had the good fortune of being at the top tertiary medical centers in the world, and I was absolutely astounded just how little we knew about pregnancy. And again, it's one of those things we could ask our OB-GYN, what represents a healthy diet? How do we optimize for our children here? And it was clear what's not healthy. And so, eating garbage all day is probably a bad thing. But what represents a healthy, optimized diet for someone who's pregnant? They really couldn't say. And that to me was astounding. And so this is the mission of the Gates Foundation, to really optimize health in these women during pregnancy, to be able to develop diagnostic tests that tell us if a child's at risk. And these are tests that can be deployed within the field. And so that mothers that are at risk and children that are at risk can be brought into medical centers then, which are not, again, close to their location of living. And so that's the basic idea. And so we've worked together with the Bill and Melinda Gates Foundation on a number of really exciting projects where they're doing large scale collections of biological samples from around the world, and simply just trying to understand what's a normal pregnancy. What does a healthy pregnancy look like? How do we better diagnose maternal issues at a very early stage, and then identify children who are at risk for early childhood illnesses while they're in utero, so we can help optimize their health?
That's very interesting. I could think of an experiment or a study where you take, samples from these pregnant women once a month, or maybe once every 2 or 3 months, and then just measure that and see how over time the babies, function in their environment, and just to see whether or not there's any, signals you can find. Are there any discoveries or interesting findings that you found during this process?
Yeah, so there's a number of them that are emerging, and this is really the ownership of the foundation and the many grantees throughout the world with whom they work with, who are collecting these samples and running these studies. We're simply a facilitator, a service provider for them. So I'll let them speak about the discoveries. But again, there's a number of these that are emerging where what we're finding, precisely to your point, Ray, that there are markers that appear very, very early on in pregnancy that tell us what's going to happen to the mother and what's going to happen to the child. There are markers in blood, and there's markers in breast milk. There's markers in the earliest date of what's called cord blood when the child is born, the blood that's emerging from the child, that tell us quite a bit. And it's just the case that no one's looked before. And now that we have the ability to look across very large numbers of individuals, discoveries can emerge.
I can imagine in the far future, not only will they have to, or they won't have to collect samples and send it to a lab, but you can sort of do it at home in more real time. And I think that's an amazing future where we can really improve ourselves, day by day, very effectively. So interesting. When do you think that's possible? What's the a real time in your blood sort of analysis?
Yeah, I think it's absolutely possible. And it's not and we do this today. So if you have diabetes, we do at-home glucometer monitoring where we measure glucose. We have little devices that you put on your arm that continually measure glucose, as you know. We have quite a bit of point of care testing. We saw this during COVID when we were able to do lateral flow assays and COVID testing at home, how this completely changed our ability to self-diagnose disease. And I think this is evolving very quickly. It's not a technology issue, and it's not a cultural issue around patients per se. I think if you know what to measure, you can develop the test. It's an information issue in that we just don't know what are the right markers. And so that's step one. What is the actual best diagnostic? Let's identify that singular molecule, and then let's develop that point of care testing. And I think this is exactly what people want. I know personally, being able to take ownership of our healthcare, being able to ask the question at home, I've got a cough, do I have COVID, has changed the way we think about self-medication and just self-diagnosis.
So interesting. Let's switch gears a bit. As you know, Bio helps early stage biotech companies find funding and gets them through their inflection points through the biotech journey, let's say. But I know Sapient is a little different because it's a service company, as you mentioned. But how are you seeing the biotech funding landscape? And has it affected the way you're seeking your next round of funding?
Yeah, and you know this better than I do, Ray, but the landscape and environment has completely transformed over the last 18 months. And it's really, and obviously there's many factors that have contributed to this: COVID, interest rates, et cetera, the availability of deployable capital. There's obviously an enormous amount of investment that's gone into healthcare over the last several years during the COVID pandemic, and even prior to that, not only related to COVID, but also around all sorts of healthcare: oncology, cardiovascular disease, neurodegeneration, Alzheimer's, et cetera. And the challenge with investment in the biotech sector is it's always one that's been almost binary in its success. If you're developing a drug, there's a series of raises typically that occur, and you don't know until the very end what the inherent value is of that discovery, which from an investment perspective is really quite difficult. Now, in the times of low interest rates, that's a very viable sort of investment. I think as interest rates have changed and the cost of capital have changed, that becomes a much more difficult sort of endeavor to support. And this is where I think biotech is evolving tremendously. And we're seeing this already: ideas like AI-based drug discovery, decentralized virtual biotechs. This is where much of the success is coming. And it's not necessarily because the idea is different, but rather the capital efficiency is quite a bit different. And so as we were forming Sapient, this was really at our forefront. And I don't know if it's my conservative nature or what, but the idea of having to do serial raises and not know the value of what you're developing until the very end was challenging for me. And so the way we built Sapient from an economic model is one in which we are a service provider. We have these technologies. We're providing services around analysis of biological specimens for large pharma companies, biotech organizations. We're helping them to analyze and generate this incredibly valuable data, as well as understand that data for the purposes of their drug development. And so first and foremost, we're a service organization, what's classically called a CRO, that's supporting the biopharma sphere and their drug development efforts. Because of that model, we're able to generate revenue very early on, and that revenue feeds and supports our internal R&D efforts. And so again, it's a very different model, one of which is far more sustainable. I don't want to make claims around omniscience. Certainly, we had no idea how this was going to evolve, but early on, this made a lot of sense to us, even in the midst of what I would like to call endless capital availability 2 years ago. This type of model made a lot of sense to us. That the point of a business is to be able to do things at a profit, and being able to achieve that as quickly as possible made a lot of sense to us.
So, would you say, are you seeking next round of funding? And I feel free not to answer or refrain from answering, because I know that some things like financial questions could be off topic, but are you seeking a next round this year, next year, and what are you doing to get there?
Yeah, so we're always open to conversations, and what I'll say, Ray, is even when we took a single round that sort of formed Sapient and launched us, and even through that process, we were never seeking funding, if you will. It was a somewhat of a happenstance interaction that led to our investment partner, and it was just the perfect fit for us. It made a lot of sense, and that's what sort of prompted it. And I would say that we're always open to conversations, but we don't actively require funding. So it's a very unique position because, again, we're a service organization. We're continually generating revenue. It puts us at a very different position and a very different economic mindset. Now, again, growth has been fierce and fast. We have continued to sort of develop great efficiencies, both through hardware, software, processes, et cetera. So we continue to get better. But there's always need for additional capital. Now, it is our goal that capital comes from our internal sort of service providing or offerings again. But we'll see how the future evolves here.
Where do you manufacture your hardware? I'm just wondering if there's any sort of supply chain challenges that you faced in the last few years.
Yeah, so certainly supply chain issues have been complicated. It's a little bit different because we're providing services, so we don't sell hardware. So it's a little different. We're focused here in San Diego, California, where we have our central laboratory, and we can handle now over 1 million samples a year on site for analysis and have the ability to scale approximately 10x on site. So it's very different. So what we're providing is truly access to those technologies and the data, which is the valuable component, rather than the underlying hardware. Now, there may come a time in life where in our evolution that may change, but for right now, at least, that's our model for operations.
It sounds like you have a large enough capacity to handle the samples that you're getting for now.
That's exactly right.
So can you describe some of the partnerships and some additional partnerships you have, maybe with other academia, biopharma companies, big pharma, or even government? And how have you structured those deals as well?
Yeah, absolutely. So the majority of what we do, again, is with large pharma organizations. And essentially, we serve as their bespoke service partner. The way we operate is one through a culture of collaboration, we say, where we essentially become an extension of their own chemistry and analytical groups. Now, the types of questions we're answering can vary tremendously, from groups coming to us and saying, we need to identify an early target in liver disease or in heart disease or in cancer. We want to validate this target. Can you help us do that? All the way through early phase 1, phase 2 clinical trials, where individuals may come and say, well, listen, we did this phase 1 study, but we're seeing heterogeneity in response. Can you help us understand why? Can you help us identify a marker in vivo in the human that tells us that our drug is doing what we think it's doing? All the way through phase 3, phase 4, sort of large epidemiologic studies. So we support the entire spectrum of drug development from the very, very earliest stages through toxicology, through phase 1, phase 2, up through phase 3, phase 4 types of studies. Now, again, that represents the majority of what we do. There are a number of academics also with whom we work. One of our sort of most prominent academic relationships is Cedars-Sinai Medical Center here in Los Angeles, where we're able to bring in tens of thousands of samples annually for analysis. Very similar to a relationship to what we have with the Gates Foundation, where we're a service provider for them, but given their academic and nonprofit status, we're able to provide services at a slightly different sort of cost point.
That's great. It's really interesting. Thanks for sharing that. And Mo, this has actually been a really great conversation. I feel like we've learned a lot about diagnostics, the future of diagnostics, a bit about funding and the landscape there. Do you have any other things you want to share with the community? Otherwise, we can sort of wrap up here.
Yeah, absolutely, right. The last thing I'll say is that there's no one path to success. And that's a lesson we've learned. And there's no one right answer. Obviously, there's formulas and there's typical ways of operating. But as you've taught me in the past, oftentimes success comes from individuals that do things a little bit differently, from a slightly different mindset. And that's the sort of last lesson I would leave some of your listeners with, that there's tremendous opportunities in biotech for early stage organizations. The ceiling is quite high as to what we need to do and where we will go over the next decade. And so for individuals interested in getting into this space, taking that first jump is really the hardest part.
Very well said. Dr. Mo Jain, thank you very much for your time today.
Thank you so much, Ray. I appreciate it.